All right. Thank you, everybody, for joining us today for the University of Minnesota's Conservation Sciences Seminar. It's my distinct pleasure to welcome our speaker today, Dr. Michael Phelps. He is an assistant professor in the Department of Animal Sciences at Washington State University and helps direct the WSU Thorgard Center for Salmonid Physiology and Genomics Research. He has a bachelor's degree in cellular and marine biology from Western Washington University, a PhD in environmental science and aquaculture from the University of Rhode Island's Department of Fisheries, Animal, and Veterinary Science, and has postdoctoral experience in muscle stem cell biology and genome editing technology from the University of Washington. His lab currently studies the molecular physiology of aquatic organisms with a special focus on the molecular mechanisms underlying fitness traits in Pacific salmonids. And on a more personal note, for those of you wondering, Mike is my cousin, and I can assure you that his passion for salmon go well beyond the lab bench, and he can catch these fish as well as he can sequence them. So I'm very excited and proud to have him here joining us today. Before I turn it over to Dr. Phelps, one last comment. If we have time at the end for questions, Mike is open to having you just unmute yourself at the end, or along the way, you can put them in the chat box and we can read them to him. So without further ado, Mike, I'll turn it over to you.
All right, great. Thank you very much. This is a fun opportunity to be able to talk some science here. Okay, so today I'm going to my lab is a little unorthodox in the approaches that we use for conservation, but I hope that we'll be able to create some dialogue and give new ideas of potential applications. And I'm going to specifically talk about the work that we use using Crispr genome editing technology and how we're applying that to salmonid conservation in the Pacific Northwest. Now, my lab does a lot of different types of work. A lot of it is mostly focused around Pacific salmon. Some of the big bulk of what we do is just basic biology and physiology of the animals. We also do some ecological genomics work to try to identify genes and traits, and then also have a biotechnology component where we're developing new tools to be able to further salmon conservation and biology. So as Nick mentioned, I started a little bit over a year and a half ago, and we've started a center at the Washington State University called the Thorgard Center for Salmonid Physiology and Genomics. And this center is really just focused on advancing fundamental knowledge of salmonids and for use in conservation and aquaculture. And in this talk, I'm going to focus on 4 aspects of how we're trying to address salmon conservation using Crispr genome editing technology. We're going to talk about how we use this for detecting species, techniques called genetic barcoding, using gene therapy on wild conservation applications. And then finally, we're going to touch on more controversial issues, which is facilitated adaptation. So before I get started, I want to give a brief background. I always talk about CRISPR because to me, it's fascinating, the basic biology of what CRISPR is and how we've been able to develop it into this multifunctional tool. CRISPR is found in the vast majority of prokaryotic organisms. It's found in 50% of all bacteria, and 90% of archaea have some type of CRISPR system in them. And what it really is an acquired immune system for bacteria to fight viruses. So when a virus invades a prokaryotic cell, the cell can essentially capture, if it survives that infection, capture a portion of that viral genome and implant it into its own genome. Now, it implants it into a region, the CRISPR region of its genome, and that stands for clustered, regularly interspaced, short palindromic repeats. It's a really long mouthful. But essentially, it was this strange sequence back in the 80s that they didn't know why it was present in prokaryotic cells. And it was essentially these repeat sequences. In each repeat, space between them is a viral genome, a viral sequence. And what the cell does is transcribe this CRISPR locus into RNA, and then cleaves that RNA into small little RNA molecules. And then an enzyme, a nuclease, can bind onto that RNA molecule and search the cell constantly, looking for a new virus that's invading that cell. And if it recognizes it, it can cleave that virus before it can infect the cell. So that's how it uses CRISPR as an acquired immune system to be able to prevent future infections. And it's been amazing that we've been able to actually capture this technology and be able to use it for our purposes, because the enzymes and the process works in almost any cell, whether it's a prokaryotic cell, a plant cell, animal cell, anything. And all we need to do is have two components. We need to have that nuclease, that enzyme that can go in and cut DNA. And then we can program that nuclease to go to specific regions in the genome with a small piece of RNA that we can synthesize and literally order from any company like we would any standard oligonucleotide. And so for the most common CRISPR systems, this is about 100 base pair fragment. The part that actually recognizes the genome is only about 20 base pairs. And so this provides us a vast ability to be able to go in and create specific edits in cells, because we can direct the CRISPR to go and cut those regions. And once we've cut those regions, then we can trick the cell to being able to repair it in a certain way. So this is kind of the basics of CRISPR and how we use it. Now, this is not a new idea, but everyone's realizing that CRISPR is everywhere now. It's in biomedical research, it's in agricultural research, it's in basic biology. And so there's lots of people engaging in the question of whether CRISPR could also be used for fields such as conservation. And so that's what we're really going to focus on today. Now, I may disappoint some people because I'm not actually going to talk about gene drives, but that is one hot topic of which CRISPR is used for. And it's mostly because my lab hasn't really done a lot of work with gene drives, and I think there may be better alternatives than some of these high-risk gene drives that have been proposed for being able to combat invasive species, which are a major problem, obviously. But I'm happy to talk about gene drives at the end if there's any questions about that. We definitely, it's on our radar, but we haven't really started to think about producing gene drives for invasive species in Washington. What I'm going to talk about is Pacific salmon and how we can apply some of these tools to their conservation. So this is my field or whatever, Washington State. And in Washington State, we really kind of focus on 3 geographical areas that we kind of segment out as specific salmon habitats that we can kind of lump together. One of them is all the streams and rivers that flow into the Puget Sound, the streams and rivers that flow to the coastal regions, and then all the tributaries to the Columbia River are kind of our 3 main segments that we kind of focus on when we're thinking about salmon conservation in Washington. And part of the reason why we have to separate those groups is because salmon are not breeding, they're not interbreeding between tributaries. And so because of that, the salmon are actually broken down into evolutionary significant units. And each one of these has to be managed independently of the other groups because of their unique genetics and their fact that they do not crossbreed. And so as far as the Endangered Species Act is concerned, or any kind of regulation, these are the evolutionary significant units for salmonids that have to be managed independently of each other. So we think of them as different distinct groups. So one thing that many of you may not know is that while we have an active fishery for salmon in Washington State, it is heavily supported by hatchery stocked fish. Hatchery fish actually make up 80 to 90% of all the salmon that are caught in Washington waters. We produce millions of them every year. They have this is the largest salmon hatchery system in the world. And here we have our basic hatchery maps for management through the Department of Fish and Wildlife. But producing all those fish can have impacts on the wild stocks themselves, because we know we're gaining a more appreciation for the fact that hatchery fish can actually impact either interbreeding with them or reduce the fitness of wild fish. So it's not a complete solution to this problem to just put more fish in the rivers. So one thing that biologists are particularly interested in is being able to identify which fish are hatchery fish and which fish are wild fish to help aid in that management, as well as we can use hatchery fish as a proxy for approximate abundance of wild fish that may be in that same tributary or area. And so there's a huge effort in Washington State, at least, to mark hatchery fish so that when they come back, we can identify where they came from, and that helps with management. So one of the most used ways is essentially coded wire tags, and this is common all across the country and around the world, but it's heavily used in Pacific salmon. Essentially, in order to a small wire tag is put into their nose, and usually we clip the adipose fin to remove it so that we can identify hatchery origin fish from wild fish. And then if one of these hatchery fishes is collected and we can identify that they have a metal tag in their nose, then a biologist goes through and sections out the nose of a fish to actually identify where that fish came from based off of the code on the tag. So recently there's been a push to try to maybe see if we can do this not necessarily with physical tags, but with genetics to try to identify where fish are coming from based off of their genetics or their genetic variation instead of needing tags. And there's a big effort pushed through by the Columbia River Intertribal Fish Commission to essentially perform amplicon sequencing on large numbers, thousands of salmon every single year to identify their origin and where they came from. So this brings us to the point of how we could maybe use genome editing technology to help enhance some of these management of salmon and to improve their conservation in Washington. And this is some of the work that my lab is starting to do right now to try to be able to bring this to reality. So the first thing I'm going to talk about is using genome editing for species detection. Now, many of you are probably familiar with environmental DNA. It's a booming area of research, with the idea being that we don't have to actually sample fish to be able to determine which fish are in a specific environment. And this is a really powerful tool because we no longer have to spend all the effort to catch them. So this is one example of flow through for a basic environmental DNA. Essentially, we would extract DNA from the environment. In this case, it would be a water sample, filter that water sample, extract the DNA, and then run PCR on that sample to be able to detect the presence or absence of a specific species in that environment. And this will give us identify whether specific species are present or in that river system or not. And this has been widely used. It's not an uncommon technique to be applied today, and there's many applications that are using it. But it does require some specialized tools, particularly the ability to do polymerase chain reaction, which can limit its ability to be used in the field directly on a site. And so there's some new alternatives to using CRISPR technology that we think we can use for environmental DNA. So what CRISPR allows us to do is essentially have a detection device which can produce a signal without having to use fancy tools such as PCR machines or detection devices. Essentially, we can extract DNA, incubate it with some polymerases and the CRISPR enzymes for a little period of time, and then you can literally put this on a test strip and have positive or negative results from a flow strip. And this is commonly being used now in biomedical research for the detection of viral infectious diseases. There's a rapid test for coronavirus that uses this approach. There's a lot of other viral diseases that are also being detected in the same way. There's even several biotechnology companies that have implemented this technology as well. But the same technology that can be used to detect viruses in the environment can also be used for species detection using environmental DNA. And there's been several groups that have been able to demonstrate this in salmon as well as other species to be able to detect the presence of those fish in an environment in the river system. So how does this actually work? It essentially uses two processes. The first process is to use an isothermal amplification so that you can take a small amount of nucleic acids, either RNA or DNA, and amplify that up to a larger signal. And then by applying CRISPR to that, we use special CRISPR enzymes that are not the traditional ones that they would use in gene editing. This is an enzyme called Cas12a. There's another one called Cas13 that essentially, when they bind their target sequence, they activate and they look for single-stranded DNA or RNA around them and start degrading all of that single-stranded DNA or RNA in the sample. And so we can put a probe in the mix that is either a fluorescent probe that has a fluorescent quencher tag on it, or a biotinylated probe that can bind to a flow strip that we can then use to detect the amplification that CRISPR determines. And so we can use this either with a traditional fluorescence detection or in using it with a flow strip that I showed you. So essentially what that is combining amplification with CRISPR detection, and we don't need any specialized equipment. You can use this with a single strip. So that allows us to now be able to take this out into the field if we want to try to use that for management opportunities. So currently we are working on developing several of these different assays to detect different species. We're working on detecting 7 different salmon species and 2 fish pathogens currently. And then another approach that we're using is to be able to use this to determine sex determination in fish that have defined sex determining regions such as salmon, to be able to identify fish that aren't sexually mature and what their gender is. And this can have applications in sorting fish when they come back to spawn, or in sorting juveniles, if you're interested in understanding what the distribution of sexes are. So here's some of the data that we have for detecting this Renibacterium salmonarium. This is a bacterium that's used, or that causes bacterial kidney disease. And so this is just showing that we can detect that pathogen using this CRISPR approach. So this is a way that we can use CRISPR that has nothing to do with actually modifying animals at all. And instead, we can use this as a tool to be able to detect the presence of species, an alternative to current eDNA technologies. Okay, but that brings us to the question of like, a lot of people, the possibility of using CRISPR on actual organisms themselves. And that's kind of where I'm going to focus for the rest of the talk, because this is where people mostly consider potential applications for CRISPR. And one thing that we're working on is something called genetic barcoding. And the idea is that we could, if we can put a mark in the genome of a hatchery fish, we could replace the ability to need to use coded wire tags to identify where those fish come from. And we can use that genetic tag as a mark for where the hatchery of origin of those fish came from, and use this as a management tool. So how does this work? How would a CRISPR barcode work? Essentially, we would take a region of the genome, and in this case, we identify a region of the genome that's as far away from any kind of gene as possible. We want to kind of identify a desert in the genome that we believe has very little or if any impact on genes themselves, and to just put a small base pair change in that region that could occur naturally through breeding, but instead we put it directly where we want it to be so that it's a barcoded sequence. So you can imagine here, if we have our gene sequence there, if this is our barcode sequence in that region that we know is where the barcode is, if we have hatchery 1, we can add the CRISPR enzyme in and add essentially a base pair change at that location to identify that this fish came from hatchery number 1. And then for hatchery number 2, we can have the fish barcoded at another location. So now we can identify with a simple PCR and sequencing where those fish have originally came from. So the idea is to be able to mark hatcheries from specific stream types with a single area, so we don't have to do this widespread genomics approaches that we're doing now. This would dramatically cut down costs of being able to identify the origin of the fish. And because genetic barcoding is not lethal, we don't need to essentially kill the fish and cut off their noses and find the coded wire tags anymore. We can do this from a DNA sample, either by fin clips or environmental DNA. We could do this in the middle of the ocean to check abundance of fish on specific stocks based off of their hatchery abundance, if we can just genetically barcode a region of the hatchery fish to be able to tell where they came from. So we do have a pilot study to see how this would work, but we're not using salmon for the pilot study, mostly because to really test this, we have to see how this works over several generations. And so right now we have a pilot study to use zebrafish to be able to test genetic barcoding in the zebrafish. And essentially we've genetically barcoded 2 different unique stocks of zebrafish. And we're combining those after we get the populations up to the level that we need them to. Now we're going to do a mock environmental study where we can sample individuals from that population to get population estimates based off of how many fish we can determine that were sampled from the total population. And then we want to be able to run models to be able to see how this would work in a potential management setting. Now, some of this work is in collaboration with Wes Larson, as well as Garrett McKinley. Wes Larson is at NOAA now, but he used to be at USGS and Sandpoint. And Garrett McKinley used to be at the University of Washington, and now he's at Department of Fish and Wildlife. We're working on this project together to try to see if this is something that managers could use for salmon conservation in the future. So here is this is an example of our barcode sequence that we have in zebrafish right now. We have identified 4 different barcode locations that are potential to be used in this location. There's many more of them that could also be used, but we're spacing them out right now. And this is, again, found in the empty space of the zebrafish genome. We don't want it to be near anything. We just want to add small little pieces, small little genetic changes at those regions that, again, could occur naturally, but we're placing them in that region to be able to use as a marker for where that fish came from. And the way this essentially right now, we have to do this through traditional gene editing or genetic engineering approaches by injecting eggs, but we essentially injected zebrafish eggs. Those aren't zebrafish eggs, those are trout eggs. We injected zebrafish eggs with CRISPR cassettes to essentially insert a mutation into one of those barcode sequences. And then we can do amplicon sequencing of that whole region to identify where that fish came from based off of its mutation. So now this is only proof of principle, and doing that approach by injecting the eggs is not going to be feasible on a population level. And so there's been a large interest. There's actually a large interest in the Pacific Northwest community to potentially apply this approach. In fact, this type of idea has been applied in the past by selecting individuals with specific genotypes for certain stream systems so that you could genetically identify where they came from. But now we have a tool to be able to do this much easier. Some of the things that they're really interested in is possibility of not just identifying where that fish came from, but also year classes by marking multiple loci within the barcode for the different stocks. So these are something that is on the minds of fisheries managers, but is still on the forefront of being able to think about how this may be applied. And so one of the tools that we're using to be able to maybe apply this at a larger scale is taken directly from biomedical research, and that is gene therapy. Now, most people would never think of using gene therapy on a wild animal for a conservation purpose, but it's completely applicable to being used on non-human species. And essentially, gene therapy mostly uses viral vectors to deliver the genetic material, in this case, CRISPR, to cells. Now, this is, if we were to apply this in the context of genetic barcoding, it may be possible to apply gene therapy to sperm from salmon or other fish to be able to edit them at large scale in that pool. And then we would be able to pass that on through that next generation. But there's many more applications for potential gene therapy applications in aquatic organisms. In fact, our lab is mostly interested in not for use in genetic barcoding, but mostly for basic science, understanding how genes work and how that can tell us more about the biology of salmon. But we can also use them for something called somatic genetic engineering, which I'll talk about in just a second. And we do have a funded grant to be able to see how these gene therapy viruses could be used to actually combat viral diseases, essentially having viruses seek out and destroy other viruses. It's kind of a radical idea, but that's something that we're using them for right now. So what we're developing in the lab right now is exactly like would be a traditional gene therapy type application that you would use in humans for medical treatments, except for we use this using a fish virus. So for this work, we're actually using the VHS virus, but it's not the VHS virus that you would imagine. This virus is harmless. We're just using it as a delivery vehicle. So the way we do this is essentially take the genome of VHS. Here's the genome of VHS with all its pathogenic genes. And we can strip them all out of the genome, just take them all out, or at least most of them out, and produce them in cells in recombinant form so that we can create the virons to be able to deliver this CRISPR to actual cells themselves. And this is the same technology that's used for gene therapy in humans, except for we use different viruses. And in fact, one of the most potent gene therapy viruses used in humans now is derived from the HIV virus, but it's harmless because we've taken the genes out of it. In fact, 2 of the COVID vaccines that are going in circulation now are gene therapy vectors, just like this one would be. So if we have this gene therapy technology, now we can use it for all those applications that I talked about. And I'll go through how we can use that for doing a little bit more of advanced CRISPR work going forward. All right, so I'm going to touch on the final topic that I'm going into here. And this is something that is more controversial than some of the other approaches that I was talking about, because now we're trying to actually change organisms and how they are able to adapt to their environmental sequences. And so the term that we use for this is facilitated adaptation. It's essentially boosting the evolution of a species to try to save it from some type of threat. And this is actually pretty far advanced, this idea of using facilitated adaptation to fight things such as coral bleaching. In fact, there's been a lot of publicity on this with corals right now because there's lots of work that are looking at the possibility of being able to produce coral hybrids by taking corals that are resistant to bleaching or warm water temperatures and crossing them with corals that are sensitive to bleaching so that they can have reef systems that can survive what they believe is a catastrophic event that's occurring, that is not only occurring, but is scheduled to get worse throughout the world, to be able to preserve coral reefs in regions where they're being destroyed right now. And so these would be essentially replacing the corals at that site with hybrids that could survive bleaching events. So how does this work? This is actually called assisted. This is different than facilitated adaptation. This is called assisted gene flow. So we're crossing 2 animals together to try to get that. We're providing gene flow to be able to have the benefits of one organism into another one. So you could imagine if we had, I could put corals up here. We could put any type of animal up in this environment, but imagine similar scenarios under which you have a tolerant population and a threatened population, and we want to try to see if we can save the threatened population. Assisted gene flow essentially would mean that we're going to cross or implant tolerant resistant animals into that environment so that they can breed together, and hopefully the hybrids between those 2 populations, some of them will have the trait that was given to them, and they will be able to survive in that environment where the threatened animals may not be able to. So, but if you think about this, the resulting animal that's there, either a coral, fish, anything, is not the same organism that was originally in that threatened population. It's now a mixture of the 2 strains. And if the tolerant population was more, had a higher fitness and was more well adapted, it may actually take over the threatened population. And while you still would have salmon or you still would have corals there, it's not the same species that was there, or it's not the same population genetics that was there originally. So facilitated adaptation works a little bit differently. And this is where using gene editing to try to do this process. And we still use tolerant populations and threatened populations. But in this case, we're not actually breeding the 2 together. What we're doing is we're taking a single trait from the tolerant stock, and we're putting it into the threatened population. And so this could be as small as a single base pair change at the right location in the genome. And all we have to do is transplant that to there. It could occur naturally through assisted gene flow or natural breeding processes through adaptation. But what the process of getting that acquired mutation would lead to many more genetic changes across the genome of that animal. And so in this case, facilitated adaptation, the animal at the end looks very, very similar to the threatened animal at the beginning. It just has slight changes at specific regions to be able to give that animal a boost in adaptation. So this is kind of the difference between those two approaches. And we're really dealing with species that we're really worried about collapsing. So one approach that this is kind of scary when you start thinking about permanently genetically modifying wild organisms to try to enhance their ability to survive. And so one way around this is not to actually modify their genomes at all, but to use the approaches such as gene therapy approaches using viruses to be able to add those traits into stocks of fish or stocks of threatened animals, but that won't actually alter their genome going forward. This would allow them to get through a threatened period of time without having to actually permanently modify them. And you can think about this. This is why we use gene therapy in humans, because it's unethical to genetically modify humans so that they'll pass it on to their offspring. Instead, we always just think, okay, well, if we're going to use this for therapy, if someone has a terrible genetic condition, we can use gene therapy to fix that genetic condition in that one organism, but then their offspring will be normal again. And so the same approach can be applied to conservation by temporarily fixing populations of fish before they can, without having to permanently alter them. So this is an alternative. This is called somatic gene editing or somatic genetic engineering, because we're not changing the germ cells. Okay, so these are obviously fairly radical ideas as far as like extreme conditions to how we would maybe intervene in the case of a species that was going extinct. And lucky for us, or it's not lucky for us, but there's lots of species that are going extinct every day and all the time. But most of those are fairly unknown species and don't have major industries or major investment. But here's my example of the Chinook salmon in Washington state. It's been hypothesized that certain populations, many of the southern populations of Chinook salmon, will not be able to survive in 30 years with the increases in climate change. So am I willing to accept the loss of Chinook salmon in Washington or other areas? It's something that I personally don't want. And what kind of steps would I go to be able to do that? Now, if I want to bring that a little closer to home, imagine going out and trying to fish for a walleye, but having the same kind of effect with something devastating that would cause them to collapse. What would we be willing to do to bring them back? And now those are just two examples that aren't really relevant right now, but for many species, that is actually what's going on at that place. And so some of them are at a crossroads where we have to make a decision whether preserving that species in the ecosystem is more important than preserving their genetic purity of what we believe those fish should be, or those animals are, whether they're pure wild animals or not. And this is an important distinction, and everyone can have their own opinion on where that should be. But I would make the argument that as soon as we get to the point where we start captively breeding small populations of animals, or we start thinking about processes such as assisted gene flow, like with the corals, where we're going to breed hybrids to try to get them to be in the environment, that those have already dramatically affected the genetic diversity of those stocks. And that alternative methods such as using CRISPR might be another approach to be able to enhance those. So this was a paper that we wrote, and I had to bring this slide in here because it gets us thinking about this topic of balancing the maintenance of genetic diversity in wild fish with the possible use of genome editing to be able to enhance the ability of those animals to adapt to conditions that are out of their control. And so we kind of created this framework where the goal is to try to not do any type of genetic modification in animals, But if animals begin to be threatened, that it could become an option based off of how threatened those animals are and how impacted their genetics already is. So in animals that are just beginning to be threatened or vulnerable, approaches such as genetic barcoding or somatic gene editing that aren't going to cause major changes in the genome are something that would be preferred and something that would maybe be considered. And you wouldn't ever consider doing things to facilitate adaptation until you're really concerned about the loss of those animals in an environment and whether you could actually be able to help preserve them through those approaches, because their genome has probably been seriously bottlenecked already, and any kind of changes would not necessarily impact it further. So these are just kind of an idea of could gene editing be used to help species that are under threat. But there's a big problem here, and I always get this question all the time, especially in the case of Chinook salmon and thermal tolerance. There's a lot of people that are really scared about the loss of Chinook salmon in Washington, and they always say, well, can't you just make a thermal tolerant resistant Chinook salmon? And I say, yes, but tell me what to edit, because the technology is there where we can create, we could do the facilitated adaptation. We could transfer alleles from one population to the next. But what are those alleles that actually do this? This is the major problem as far as approaches to be able to use this in the future. It's not the technology, it's the biology that we need to understand, which we don't have a very good understanding of right now. And so this is why a lot of my lab focuses on basic science to understand the biology of organisms, if we were ever to use any of our knowledge to be able to help them in conservation. So I'm just going to go through one example of how we are using CRISPR for basic science in our lab. We're really interested in this pathway called the activin receptor signaling pathway. This pathway is incredibly important in the biology of all vertebrates and even some invertebrates as well. It's expressed in almost every single tissue in fish. It has major roles in skeletal muscle development, embryonic development, and reproduction. So but we don't have an idea of how this pathway is really working overall. And so some of the work that we've been doing recently is to try to map this pathway through gene expression analysis, where we use a technology called nanostring technology that essentially uses little barcodes to be able to identify the expression of hundreds of genes simultaneously from a single sample. And so we've taken essentially we use rainbow trout as a model system in this work, but we're essentially looking at the gene expression of every single member of this entire pathway. It's like 50 plus genes, and across 30 plus different tissues and developmental stages to see how they're regulated. And that will give us an idea for guiding future experiments to understand the function of those genes. So here's just some of the data. We're creating a whole atlas, a gene expression atlas of this pathway. We have intercellular signaling proteins, receptors, ligands, inhibitors. On the y-axis here is just their expression level. But now we're able to look at the entire major picture of this pathway of a lot of different genes over a lot of different tissues. And we can start to build consensus of how this pathway is working to regulate the growth and development of specific tissues. So here are some heat maps just picked out of specific genes. In fish, these types of studies are complex because many of these genes are duplicated through some genome duplication events that occurred in fish. And so we may have 2 or 4 copies of many of these genes, and how they've evolved different functions over different tissues is something that we need to know if we were ever going to do any kind of genetic manipulation of these animals in the future. So here we got green that expresses high expression, red low expression, and we can see that things have within all these different genes, there's a variety of expression over different tissues. And we can use that expression to start to map how that pathway actually works, what kind of receptors specific ligands are working with, what kind of inhibitors are most likely interacting with those based off of their expression pattern. And it gives us kind of a whole picture of this pathway. And it allows us to identify genes that we never thought were important in specific tissues. One of these ones up here, activin beta C, INH beta C, that you can see on the top there. This was a completely surprise gene that we did not expect to be expressed. This one was expressed in muscle tissue, extremely high levels. It has very little literature on it at all, and was a complete surprise. But now that we have this atlas, we can identify some of these factors to be able to try to understand their effect further. But that kind of leaves us with a problem, and that is we can make correlations with general pathways and gene expression to identify how some of these pathways work. But we need to, at the end of the day, we need to get down and do the dirty work to understand the function of genes. This is the molecular physiology to identify how they work in the animals themselves. And this is where we use gene editing and CRISPR to be able to test the function of those genes through gene knockouts or different reporter assays that we can do in them. So here's an example of some of the fish in this pathway that we're creating mutants for to see what their functions are. To create a mutant, we essentially inject the eggs with CRISPR constructs, except for this time we're not, unlike genetic barcoding, where we're targeting an open expanse of the genome. Here we're targeting the actual genes themselves to see what happens when those genes are no longer functional in fish. And to add as a marker to show that the fish are actually edited, we usually also edit a pigment gene as well. So you can see that the name of the pigment gene here is SLC45A2. These fish were edited at both this activin 1A gene as well as this pigment gene. And the fish that have the loss of pigment are the ones that were gene edited. We can sort those out of the population quickly to identify gene edited fish from non-edited fish really quickly. Now, this process is not 100%, so you could the fish below is actually edited as well, but it's only edited in a portion of its cells. And so we can, these are what we call mosaic animals that have partial editing overall. But we're using these as not as a system to try to use for conservation, but to understand the biology of this really, really important pathway that could be involved in conservation in the future, or any type of application that we may want to know more about this genetic pathway. So essentially the whole picture here is, I don't want you to come home with the idea that CRISPR is going to solve conservation, because it's not. It's not a solution to conservation issues. It essentially is a new tool that we may be able to use to improve the efforts that are already going on. So it's not going to replace hatchery rearing of fish or any species. It's not going to replace habitat restoration. It's not going to replace general fisheries management, but it can be used as a tool to be able to help further advance those approaches. But we can't do any of it without knowing the biology of those fish, because when we start to get down to the level of having to understand what we would change if we were going to change anything, we need to know the biology of those organisms. So that's kind of the big take home that I would say from this talk, is that CRISPR is not going to act alone, and that we need to know more about the organisms themselves in order to do this process. So this is my last slide. I just wanted to acknowledge some of the members of my lab. I got 2 grad students, Jasmine and Alex, and then we'll probably have 2 more that'll come on this year, as well as a number of undergrads that are working in the lab right now, and just in some of my funding sources. But I would be happy to ask any aspect of this talk. I would love to answer any questions that people have about some of the approaches that we're using.
All right, thank you very much, Mike. Virtual round of applause for you. Great talk. Folks, feel free to unmute yourself or drop your questions in the chat box and we can go through them there. I'll just wait a second to see if somebody wants to jump in quick.
Hey, I've got a question actually. This is Isaiah. I'm a Nick Phelps grad student. I'm curious about the eDNA detection. So there's like environmental sources, obviously, of pathogens, and there's reservoirs that we don't know about for some of these pathogens. And I'm wondering, is there anything that you guys are working on to like kind of determine when a pathogen's actually maybe the virulent type, or whether it's actually originating from the host that you're curious about? if there's anything like going on like that where you can kind of interpret that information beyond just positive, negative, especially for pathogens.
Yeah. So that's a great question, and I think that is one of the limitations with like the traditional eDNA detection approaches. And it's no different for the CRISPR. The CRISPR is essentially just, I mean, you could design it if you really knew, you could design it to target specific sequences. It is more amenable to being able to detect small base pair changes than a traditional qPCR would be, because you can place the CRISPR to detect single base pair differences. In fact, some of the work that used it to identify different species can actually detect single change differences between species. But it's still going to give you, most of the cases, presence or absence. And I think we are working on other approaches, mostly using things like nanopore sequencing to be able to identify bigger pieces of the DNA that you may be more applicable to being able to identify the differences between virulent and non-virulent strains. I think DNA sequencing is probably, or just sequencing in general is probably a more applicable approach to do that. We're trying to use some of those long read sequencing approaches to be able to do with eDNA. Essentially, it's still at the exploratory phase now. We're trying to see if we can identify not just presence or absence of animals, but also abundance and distribution of animals. If we can get a big enough region with enough genetic variation in it, maybe we can tell how many animals are in that sample and where they come from. So you could do the same thing with pathogens for sure.
Thanks.
All right. Go ahead, James.
Okay, that was a great talk, Michael. Thanks a lot. My kids were watching this with me during lunch, and their minds were blown too. My question is related to this CRISPR conservation and that sort of, I can't remember what you call it, the assisted like augmentation.
Facilitated adaptation.
Facilitated adaptation, right? So you had mentioned how like you could use this and like it was kind of controversial for obvious reasons, but you would use it like, okay, in these places where the species have really important ecological conservation, you know, that like they're part of a bigger picture and you can't stand to get rid of them.
Yeah.
But it seems like, you know, a lot of species that are on the brink of needing conservation or that we sort of recognize as being endangered may no longer play a large role in the ecosystem precisely because they're endangered. And that the time to do that might be much earlier. In which case, how do you decide, like, when is the threshold? It seems like it might be too late to do it at some point and too dangerous to do it early. And what is the knowledge that's sort of going into that discussion?
Yeah, that is an amazing question. Yeah, that is great. It brings me back to my work over at the University of Rhode Island, because coming from Washington State, we would get runs of, like thousands of fish every year from salmon, but in New England, they would be some of the conservation hatcheries for Atlantic salmon, we get 5 returning fish. And I would be like, that stock is extinct as far as I'm concerned. But there was no longer a niche for Atlantic salmon in that environment. It was replaced by striped bass or something else. And so you're totally right. Their opportunity might be lost for sure. There is other examples, like some of the really heavily selectively bred, like captively bred populations, like condors, where they maybe have, they could get boosted, but they have some kind of genetic condition because they're so inbred that if you could just get over that genetic condition, maybe they could help, populate more. But I think that's a great approach. Where's that line? This is an open debate. This is kind of new in that field. Where would we be willing to go to be able to try to do that is an important question. I don't have the answer. I'm not an I don't do ethics, but this is something to really consider. And this conversation is going on not just in conservation, but also in agriculture, of where is the line that people are willing to go as far as having genetically engineered animals in their lives, for sure. So I don't have a good answer, unfortunately, but you're right. Like there's probably a good window in there that we may be starting to think about these. But the challenge really is what do you even change to try to get something to be adapted? It's easier when you just breed them together, because you don't need to know what you're changing. You just know something has a trait you want and something doesn't have it, and you just breed them together and hope for the best. With gene editing, you really need to be guided and know exactly what you're doing. Yeah.
All right, Mike, we have a couple of questions here in the chat box. First one's from Wes. In the cases of assisted gene flow and facilitated adaptation, as in corals or salmon, how are you addressing concerns of loss of local adaptation and intraspecific genetic differentiation? Or at this point in conservation of salmon, are we beyond conserving such populations?
So in salmon, we're not to the point where we're thinking about doing facilitated adaptation yet. We're considering. There's been some talk about assisted gene flow and doing that, but I agree with you. I think assisted gene flow has some major potentials to lose local adaptation of that population. Just because a stock was heat tolerant does not mean that it has the same adaptations to the population that it's replacing. And so you may have a completely loss in fitness. And so I agree that assisted gene flow may have a challenge of having to meet those things. With facilitated adaptation, in theory, if you could identify the exact trait to transfer, it's possible that most of the local adaptation traits would remain in that species, but that's not a guarantee, obviously. These are really deep questions. I think there was another part of that question, or I think did I get that whole thing?
I think you captured it, but Wes, if there's anything else you got there, just jump in or put it in the chat. All right, another question from Ray here in the chat box. Can you suggest 1 or 2 courses our fisheries undergrad should take to be prepared to use or evaluate this technology in the future?
Ooh, that's a good one. We don't even have any at our university either. As far I'm teaching a CRISPR course, but we don't get into the ethics of it. I think it's something that people should probably get around to being able to be introduced to. I wish we had more of those types of courses. I know we do have a biotechnology ethicist on campus, and it would be interesting to have them produce some type of material on something like this. But I don't know what kind of courses we would take. It's kind of tough because the way biology is set up now is you have the practitioners like me that are actually doing the editing, but we're somewhat disconnected from the bigger picture of things, and we need to be more connected as far as engaging with people that are going to help make these final decisions. Because at the end of the day, my research is mostly basic biology bench work that's not going to, it may have applications else down the line, but we're providing the basic science that would then be used to further some other agenda. So I wish I had a good answer for that one. I don't know even what kind of classes I would offer my students to be able to get them prepared for something like that, for sure. But it's necessary. Absolutely.
Hey, Michael, had another question here for you. This is Isaiah again. I was kind of curious. So I know that I actually saw a paper about like facilitated adaptation for like I think it's Hawaii birds and like they're they're really susceptible to malaria, but that's something that you could make it. You pretty easily change a gene. Like someone could come to you with that gene and say this is the gene to change. And so I think there's a lot of examples of that, especially when it comes to like animals that are endangered being not resistant to certain pathogens. But I'm kind of curious, and this is a question I ask myself a lot about my work, is what do you think are like the steps that need to happen for something like this to actually be implemented? Like in your dream world, like what do laws need to change first? Does like this need to be tried in like a island setting, or like where does this how does this like break through into reality?
Well, I mean, I think for the example of gene drives, a lot of that discussion has kind of progressed further than some of these adaptation type approaches, because they're actually considered doing this in isolated environments, that type of thing. Agriculture also, this is way farther advanced as well, as far as looking at approval to try to do these things in agricultural animals. And at least in fish, there's always the opportunity that those fish could get out. So there's more conversations on that. I think the idea is, you're going to start with proof of principle studies to show that the technology could work, and baby steps up from there. At least for the Department of Fish and Wildlife here in Washington, their laws do not, in Washington state, do not exclude the use of gene-edited fish to be released into the environment. They do have an exclusion for transgenic or GMO animals, but gene-edited animals are kind of a gray area that they haven't had to address with. And that's a big gray area for a lot of governments right now, because it's not the same as the traditional transgenic organisms that were used in those, traditional, transgenic GMO animals. So they're kind of having to do this on the fly. But I think they're going to have to address this very soon, at least.
Any other questions from the crowd? Otherwise, we have one more here in the chat box. Okay, Mike. Meg is curious if, besides the conservation endpoints that you talked about here, are there other endpoints that are being discussed, maybe for more ecosystem services, like higher nutrition?
Oh, okay. So I guess, is this like improving fish in the environment or for human consumption? What are,
things that would benefit our use of the environment. So can we make them healthier for us to go and eat, for example?
Yeah, it's completely possible. It's one of those lines that, do we modify animals? I think modifying them so they can survive is an easier catch than modifying them to be better in the environment. But for the example, for disease resistance would be something that could be really applied in that aspect where, they're healthier for us. As far as, my work does a lot, also overlaps with aquaculture a lot. And the big catch there is not to improve production rate, production or anything like that. It's to improve sustainability of the fish, to make them less impactful on the environment if they get out, or if they produce lots of waste products, or have them have less diseases. Those are things that are more, I think, acceptable as far as agricultural animals go. But as far as modifying new traits in wild animals, I think that at least my point of view, or in the public perception, is that probably let the wild animals be wild unless you really need to change them to try to save them. That's kind of my at least take on that.
Awesome. Well, we're just about out of time. Thanks again, Mike. Excellent presentation. It's going to be exciting to see this technology develop and perhaps get implemented in the future, and see where that line gets drawn someday.
Absolutely.
Thank you very much. Have a good day, everybody.